titled Enhancing Lab Efficiency, AI-Driven Automation, and Data Collection. Today is presented by Steve McCoy, who's the head of sales at Labvoice, and sponsored by Astrix. So before we get into today's topic, we can go to the next slide. We'll tell you a little bit about who Astrix is, why we're sponsoring today's session. Real quick, in short, we're a consulting company. We work specifically and exclusively in technology and life sciences and all the software that goes with that. A lot of work in the R&D space, a lot of work in the laboratory informatics space, and just general kind of strategy and digital transformation in life sciences. We've been doing this now for the better part of 28 years. One of the things we're very proud of is, nearly 1,000 employees, 90% of which holding advanced science degrees. That's something we're very proud of here. We're a science-based company ourselves, helping other science-based companies. So we've been doing this since 1995, and as part of what we do, we try to bring as much education and insights into what's going on in the industry to market, which means we bring a lot of folks like our friends at Labvoice on to share their opinions and share their technologies and share where they think the industry is going. So with that, let me go ahead and introduce our speaker for today. Steve McCoy is the head of sales at Labvoice. He's been in that role for quite some time, over a decade of experience in the life science sector. Before that, he was at an organization called TetraScience, where he, did a lot of work in digital transformation across both biotech and pharma. Steve, I don't have to read your whole bio. You're welcome to kind of pick up from here. I'm going to sit back, enjoy the presentation with the audience, and I'll come back on at the end to help with questions.
All right, thank you, Kevin. I don't know if we need to go through my bio. I'm sure people did not tune in just for that. But I wanted to introduce Labvoice prior to getting into what we have today. So our talk is really focused on using AI and other emerging technologies to help streamline lab operations. And what I'll do is I'll bring you kind of through the evolution of our product over the past 6 years to give you a sense of how we tackle some of these data collection and process guidance issues that we have. A little bit of background on the company. So we were founded in 2018. We really came out of go-to-market in 2020, smartly around the time that COVID hit, which actually was somewhat of a boon for our platform, given its hands-free nature and the ability to reduce contamination and sterility. It was founded by some people that you may have heard of before. So Matt and Dave launched a company called Cytigic, which was best known for a product called Pipeline Pilot, used to break down data silos amongst life sciences, databases, and organizations. And then Fred, who is our CEO, founded HEOS, which was a cloud-based ELN back before cloud-based ELNs were a thing. Ultimately, both of these companies were acquired by Accelerus, which was then brought under the Biovia brand through an acquisition with the SO Systems. So the mission of Labvoice is really to help, as I mentioned, optimize the way that research operations and lab operations are being performed. A fair amount of time, as we know, for scientists is dedicated to administrative data tasks, collection of the data, formatting of the data, and then analyzing it. And really, it's those first two areas that we try to focus on. And although the end goal of this data collection and formatting is to get it into a digital form, we notice that there's not necessarily a lot of technology to help reduce the amount of paper and note-taking on the back of gloves and sticky notes. There's not a way to very quickly transform that data into a digital format. So we set out to introduce technology that we use in our day-to-day lives and bring that into the lab to make processes more efficient, to improve the quality of the data that's collected, as well as the quantity, and to introduce a level of compliance, which not from necessarily a GXP perspective, but really about process standardization, which has the downstream impact on experimentation. So the first thought that we had really was, well, what if we could bring an Alexa or Siri into the lab and help scientists take notes and perform calculations and set timers? And so what you see on the right-hand side here are some early versions of both the app that we have, as well as what we formerly had as called a Labvoice Hub. And surprise, surprise, it looks very similar to an Echo Dot in some ways. What we realized rather quickly, though, was scientists wanted a visual interface as a means of confirming that what they heard was accurate or what they said was captured accurately. And so we developed a mobile app. So now this digital lab assistant is available on iOS and Android devices, phones and tablets, and most commonly deployed that way. So the first iteration of the platform was really to help capture data in a tabular format. We see that still persists to this day, and a lot of the nature of work that's being done is set up is tabular in its nature, whether that is in ELNs or Excel templates or you name it. So what we have here is the first of several demos that we have, so you'll get a sense of what the app interaction is like. And this one is a very short, simple one to show off some of the features and the way that we went about capturing tabular data.
Hey, Labvoice, capture tabular data.
How many columns are in your table?
3.
Let's name the table columns. What's the name for column 1?
First name.
What type of data is in column first name?
Text.
Name of column 2?
Weight.
Data type of column weight?
Weight.
I didn't understand. Please try again.
Number.
Column 3?
Comment.
Data type?
Text.
How many rows in your table?
2.
What is the value of first name 2?
Steve.
What is the value of weight 2?
A very healthy weight.
Sorry, expecting a value of type, number. Please try again.
190.
What is the value of comment 2?
Steve is very handsome and funny.
What is the value of first name 3?
Conway.
What is the value of weight 3?
Go back.
What is the value of first name 3?
John.
What is the value of weight 3?
Skip.
What is the value of comment 3?
Thanks, John.
Thanks. The sum of weight is 190, and the mean is 95. I sent you the results by email. Goodbye, Steve.
Okay, so you got to get a sense of what an interaction is like with the digital lab assistant that we have. Now, you saw some features in there that were pretty handy. You know, some might be obvious, others might not be. For example, when I gave the weight and I gave the wise comment, it said, no, I'm expecting a number. You could see that too when I was filling out the column headers. I had already provided the category of weight, and when I gave it another weight, it said, okay, well, this has already been registered, so it must be this, and that's not one of the acceptable values for that, so we're going to reject it. Some other things in there too, like the ability to skip over answers or go back and edit what things have been added, and then some calculations done at the end there. Again, you wouldn't necessarily use this workflow, but it's meant to show off some of those capabilities. But I think what everybody can realize from watching this is that it's somewhat of a tedious way of setting up that tabular data capture. And so the next sort of evolution that we had was, well, we need a tool to help design and map to what the scientist is doing on a daily basis. I mean, forget about all of the lessons in change management that that come with adopting new technologies to totally change up the way that they would go around collecting weights and samples and having to sit there and read that out. That doesn't necessarily make sense, especially when we talk about having some of that information already available within an ELN or a spreadsheet. So what we built was a design tool, process design tool, which we creatively named the LabFlow Designer. Now I'm actually gonna do a short demo of the designer to give you a sense of how easy it is to use. Now, when you log in, it's a web-based application. you are immediately brought to your library of protocols that you have, these digitally assisted workflows that we call lab flows. I'm going to start with a blank slate. And the very first thing I'm going to do is give it an invocation phrase. So you heard previously the invocation phrase for that particular workflow was, I think it was capture tabular data. And in this case, I'm going to call it a simple weighing lab flow. Now, in all, both the user responses as well as acceptable answers, we always have the ability to introduce vocabulary and synonyms so that the human can have, or the scientist can have, a very human experience with the platform, but they're also able to write on the back end a set of standardized answers. So I might call this, weighing demo or simple weighing demo, along those lines. Then to start to build it out, what I'll do is I'm going to add a node. And now we have a wide variety of nodes and pre-built actions. You can see the list goes on quite a while here. But essentially, there's a few categories. We have these coral and teal nodes that relate to having Labvoice say something or listen for something. These blue nodes are different actions. So this is a display card, so it's displaying information. We have a benchling inventory and many other ELN integration nodes that we'll get to in a little bit, highlighting all of that. Then there's also other types of actions like scanning a barcode or creating a barcode, taking a picture, and so on. The green ones that you see are how we interact with tabular data. So in this case, something like reading table from Google Sheets, setting the column properties, adding columns, rows, and then how we export that. And then as a demo a little bit later on too, we have these star icons, which indicate some of the more AI or emerging technologies. The bedrock is how we create these small language models or GPT interfaces for customers to query. And then TextRack is some of the image recognition in OCR. So we're able to take a picture of a printed label and get all of that information off of it. And again, I'll show you some of these later on, but I'm going to start with one of the more common ones, which is getting value by voice. So this is, in this case, we're going to look for a proper set of property for it. In this case, we'll check a weight. I'm going to prompt the user, what's the weight? And you can imagine that, especially if you're doing a large amount of data collection, that can get pretty tedious hearing all of that. So I'll add in synonyms and we'll go through that. In that way, we'll cycle through and we can speed up that process. We have value types, which is how we, again, rejected that weight earlier in the previous example, but there's all different value types that we have. So things like alphanumeric codes, which are great for sample IDs or animal IDs, volumes, and we do have a weight here. I'm going to actually switch out to number because there's some features that I want to show with that. But value types are really important because it helps illustrate how we can become so accurate even when using tricky scientific terms. So we can expect a number here rather than a phrase or an alphanumeric code, something along those lines. Or even if the alphanumeric code was provided, we might only capture the numbers that were read in that process. The same thing can be set up with user responses in terms of vocabulary, bounded ranges. I'm not going to demo it here, but basically if we're looking for a weight between 10 and 50 grams, we would reject anything outside of that. So there's all sorts of ways to improve the data accuracy by using a system like this. In this case, I'm happy with doing a decimal system and I'll choose 2 decimal places. So now once we have that, I'll use the display card to give a quick visual confirmation to the user. It is a low-code interface. There is some slight coding that's needed as you learn and get trained on the designer. I myself have no coding experience, but I'm able to build rather simple workflows. I'll add. I'll throw in a yes, no, and say another weight. If they say yes, we'll loop it around. And if they say no, we'll tell the user goodbye. And then we'll end it. All right, so I will now publish this. Simple way. And then once that's available, I can walk into the lab and then access that from my mobile assistant. So now we'll watch this demo.
Hey, Labvoice. Simple weighing.
What's the weight?
8.139.
Another weight?
Yes.
What's the weight?
8.12.
Another weight?
No.
Goodbye.
All right. You can see very simple demo there. It flashed quickly, but you could see that even though I gave 3 decimal places, it converted that to 2. So again, it allows for people to have some sort of freedom when they're interacting with it. But in terms of what then is written or what is captured, it's the values that you've determined that are important ahead of running these workflows. So the next step in this process was making the connection that, look, we could become a database and save all of this information and become another data silo that maybe something like Pipeline Pilot would interact with. But really, these companies that we work with, they've invested a heavy amount into data management systems, informatics systems, and other scientific software. And oftentimes the gaps where users, the scientists, would have trouble with data capture were a small part of the entire workflow maybe that's run in their ELN or other system. So what we did was we made the system very easy to integrate with a wide variety of scientific and instrument and lab-based software so that users could have almost an improved user experience with this. If you're sitting in front of your desk or you're at your laptop at your desk and you want to edit some information in an ELN entry, by all means, use the laptop to do that. But it's when you're in the lab and your hands are in a glove box, or you're working in a biosafety cabinet, or you're handling mice in a vivarium, where some of that information update, that becomes challenging. And that's where we see the emergence of paper as temporary records, or sticky notes, or gloves, or not that everybody likes to admit it, but the memorization and hoping that by the time they get back to their desk, they have those values captured accurately. So both within the designer, there's a bunch of pre-built nodes that you're able to use for integrations, but we also have a specific web services node that allows us to read and write from any source so long as those API endpoints exist on the target end. And what users can do is actually they can then publish those integrations and save them to their organization, so that over time they can repurpose those integrations as well as any subset of a particular workflow, so that they don't have to spend each time they're building a new workflow recreating that integration. Now, I'll show, and again, another very capability heavy demo here to give you a sense of what that integration process can look like.
Hey, Labvoice. What are my tasks for today?
Steve, continue existing session?
No.
I found these events in your calendar today. Labvoice Data Strategy, Steve McCoy, Speaking Slot, Cloud Lab, HTE, and Lab Automation Conference, Update Containers in the Lab, and Steve McCoy, Roundtable, Cloud Lab, HTE, and Lab Automation Conference. Which one would you like to hear about?
Update containers in the lab.
There is an associated lab flow with the event. Do you want to start it?
Yes.
Hi, Steve. Please scan the container to update. Do you want me to read the quantity from the connected balance?
Yes.
I have updated benchling. The quantity left is low. Do you want to reorder it?
Yes.
Let me search you molecules. Which item number?
6.
How many?
6.
Send this ordering request?
Yes.
Done. I created a request in ServiceNow. Do you want to update another container?
No.
Goodbye. Which other event?
All right, so there the idea was that we could create a set of tasks for somebody to do if they had that information in the calendar. In fact, a few users of the platform have something similar. But then they're able to initiate other workflows based on some of the logic that we set up. Now, we queried information from the Benchling inventory system. That balance is actually just this balance machine we have in the cloud that's spitting out random numbers, which is how we go and update that information. And if we were to go into Benchling, we could see that that information has all been updated. Then we have integrations with e-molecules to help people place orders, either directly through the software or even creating a shopping cart list so that when they go back to their desks, all of their order information is one place if they choose to use the web app to enter that or to place that order. So that's just one example that we have of some of these integrations. As you saw on the previous slide, there's several platforms that we work with, and we can easily incorporate others if there's things that we're missing there.
Hey, Labvoice.
So now we'll get into some of the more interesting features. So over the past really year, maybe 18 months, we've gone beyond some of the traditional capabilities that we have for data capture built within the platform. Along with the emergence of ChatGPT and other tools, we felt that there was a need to have a lot of these AI tools that are readily available, again, in our day-to-day lives and brought into the lab to help speed up, the work that's being done there and lessen the burden from an administrative standpoint. So I have two demos here that I'm going to share. The first is based on a use case where we're managing samples in a cryo inventory. So the problem that the scientist was facing was whenever they had to go into one of the dewars to get out their box of samples, is that the labels on these are just simply barcoded. There's no other identifier on them. And if they're going through and scanning each one individually, it might take some time, and the thawing process could put the samples at risk. So what they've done is they've used Labvoice to very quickly illustrate to them the layout of the box, gather all of the IDs, and then they can find the sample that they're looking for. There's all sorts of logic that we can put in before and after to either register them as new entities, update them as discarded entities, and so on. But that's what you're going to see here. And it may look like a simple sort of scan, but then there's some image recognition that we put in on top of it just to confirm to the user, hey, this is what we scanned here, and here's everything that we might have missed.
Hey, Labvoice. Check a box.
Do you want to take a photo or use the one in storage?
Use the one in storage.
Thanks. Let me analyze the photo. I sent you an email with all the vial numbers and locations.
All right, so coming off of that, you can see that as you look at the image, the updated image, there's those green boxes there, again, to in case something had frost on it or the label was damaged, the scientists could always check that if they couldn't find what they're looking for. And then below, in the bottom half of the screen, was the box layout, so that, again, people can interact and find that around. Now, the next one is kind of showcases our ability to either integrate with existing GPTs and LLMs or create new ones based on specific documents provided to us by our users. So in this case, we took a bunch of process development SOPs for a particular lab, and we now make it as a way to quickly gather that information out of those SOPs, rather than having to sit at a laptop and hit Control F and search through them in hopes of finding the right answer. So what you're going to see here is just a very short demo of that chatbot style integration.
Hey, Labvoice. I need help with the procedure.
Which category?
Process development.
What would you like to know?
How do I calibrate a pH probe for a bioreactor?
To calibrate a bioreactor pH probe, connect it to the controller. Use pH 7 and pH 4 buffer solutions sequentially, allowing readings to stabilize before inputting correct values and calibrating. See screen for more information.
How do I store the pH probe?
To properly store a pH probe, partially fill its protective cap with 3 mkcl solution, immerse the electrode end, and keep it vertical in a storage clamp.
Finished.
Do you also want to ask questions about photos?
No.
So we don't have that last piece in there, but essentially what that question about photos prompt is that we can use some of this technology to take a picture of something and then ask the LLM to analyze it more or less. So we've seen applications of that for sample storage and different types of containers, even so far as diagnosing some of the clinical observations or clinical conditions that we see within, say, mice and rats that we work with. The documents themselves in that particular video also are not hyperlinked, but the ability exists to add a hyperlink to that source. So if the user is unsure of the answer, they'd be able to click the link and be brought to the SOP or the document with that information and check for themselves.
Hey, Labvoice. Hey, Labvoice.
Sorry about that. So what's an interesting outcome of using the digital lab assistants over time is that although we don't save data, anytime one of those nodes is passed through in the lab flow designer, it creates an event. And at the end of the lab flow, if the logic has not been set up otherwise, we're going to erase that information from our system. However, you can point it at something like a SharePoint list or a running CSV file, take your visualization tool of choice, and then overlay that on the data. So you can actually begin to build some dashboards and reporting and get metrics out of the interaction with the system over a period of time. So this is one such dashboard that we put for a group working in a vivarium to track their animal issues over time. And what we're seeing here is the most common issues, looking them at those chronologically. And then down here, we even have some like more interesting insights. So in this case, they're performing dosing studies. And sometimes, just again, because people are busy, technicians will actually double dose or dose a mouse that should not have been received that treatment. And what we're doing is we're actually checking against the animal history. When they give the animal ID, we'll say, yes, that's the right animal, please proceed, or no, that's incorrect. And now in the moment, that saves the animal from having, again, like the wrong dosage and impacting the study potentially. But long term, we can save those events and then we can chart them. And now we can say, begin to look at some of the way like the process operations is working around it. So I can look at this and say, what happened in December that we had so many of these warnings issued out? Is it because the animals are poorly identified, or because we have new staff and they need additional training? Those types of things and those types of insights that otherwise would just kind of go unnoticed, or at least be received only anecdotally. Now, with the lab flow designer, you can build any process that you imagine in a scientific setting. So we work in labs and vivariums, greenhouses, and even out in the field with some of the crop science partners that we have. What we found, though, is that there are certain areas and subsectors within these life sciences organizations where this particular technology delivers its ROI or its value a little bit quicker than others. So I've used a number of lab animal and sample management examples today, but really this kind of extends to all sorts of inventory as well. So disposables, consumables, various lab supplies, and even equipment. And then of course, there's the routine lab work that happens in bio and chem labs where people are doing a large amount of experimentation and capturing all that information either via paper, moving over to computer and typing it in, or even having a colleague sit there and take their notes for them. So we can end up streamlining a lot of those processes. And what's great about this is we now have templates that we can reuse, or best practices that we can implement anytime we approach a new organization that has similar needs and workflows. And where we get started, I think it's important too, is that this technology although we have experience with it in, with Alexa and Siri in our day-to-day lives, that from a scientific application, it's pretty new. So we have a strong customer success group that works with our team or with your team to effectively, manage the process where we're starting down and we're really the project managers and responsible for training and building out workflows and getting people up and running on the system, where slowly over time, we then begin to hand it off to the subject matter experts that we're working with at these organizations. Now, we're always there to support groups their entire way through, but the idea being is that Labvoice should be a self-service model, and you don't need to wait the few hours that it is to make a very easy change to a process that you're about to use. You can take control of the designer and implement that yourself. And so just to wrap up here, there's two short case studies that we have that kind of illustrate this point. And while the benefits are nice, it's really more so about the way that they went in implementing and where they've taken it. So the University of Nebraska-Lincoln started with the designer from the very get-go, and we went through the training process with them. And within 45 days, they had 3 of these workflows that were self-built with some guidance from our team, up and running and in production that they were then able to use and still use to this day, working within their vivariums. Now, on the opposite end of that, the team at Nebraska was only 4 people. We also have a partnership with Regeneron. And from when this slide was created in 9 months leading up to April 2024 or March 2024, they had been able to scale it to collect over 170,000 data points with the workflows that they've been using. In fact, they were recognized by BioIT World for some of the innovative practices that they've implemented. And the idea being is that this tool is immensely scalable. It can support a team of 4 or even hundreds of potential users as they grow. And that's where I'm going to wrap up today. I'm happy to take any questions that you have. And just quickly, I'd like to thank the Astrix group for inviting us and hosting us on this webinar today.
Great. Thank you so much, Steve. Well, let me do a couple things here real quick. First and foremost, let me get the audio recording part stopped. We typically